Bismillahirrahmanirrahim. Assalamu alaikum. You are listening to your course in charge. I hope you all are doing well. Let's start the class with the personal introduction first. I am Humaira Sarwar, working as an assistant professor in Department of Pharmaceutics, Faculty of Pharmacy. I obtained my degree of PharmD in 2010 and did MPhil in Pharmaceutics in 2016. Currently, I am a PhD scholar. I have been associated with teaching various courses offered in PharmD program, and particularly for this course, I have an experience of around 5 years. Yet, I believe that teaching is always a process of learning, and learning to the extent that never ends. I warmly welcome you all in this course of Pharmaceutical Technology, Pharm 701. As we are in a situation where face-to-face interaction is nearly impossible nowadays, leading to a monologue form of conversation, which I do not normally prefer. Well, my idea behind teaching is an interactive class where we may engage in a healthy discussion to the topic under study. So right now, it may not be possible for you people to introduce yourself and share your views, visions, and goals for your professional career. But still, we may do an experimental exercise. I will be sharing with you a document in which you have to write an introduction of yourself, express yourself honestly and as precisely as possible. That would also be a test of your writing skills. But there is a technique behind it. The document would be shared as a OneDrive link. What you have to do is to just open the link and do not download the file. Again, I would repeat that do not download the file, but rather you have to do is to open the file in your web browser and fill the columns. Now you might be thinking that why I am asking you not to download the file. The answer is very simple. The file would be available online and all the editing done would be visible to everyone having the link. This would save energy and time to download, fill and resend. But try to maintain the uniqueness as every one of you in the class would be expressing themselves in the same document. How it will help me? Well, for me it would be more convenient as I will be reading all of your introductions at the same platform. So let's start with the mechanism that will be followed for online classrooms and lecture sharing. You all have already received the introductory video on how to use HULMS. What you have to do is just to follow that tutorial. All the lectures will be uploaded with a weekly plan on HULMS and simultaneously on YouTube channel as well. What you have to do is to listen to the lecture, go through the reading material and reference books provided to you and then note down your questions. Take a screenshot of the part that you do not understand and you might need thorough explanation for better understanding, and simply share it through HULMS or on my official email ID. I will be answering to your queries combinedly at the end of the week because you would be provided a week to complete the lecture. Let's begin the introduction to the course. Before we proceed to the introduction, it is better to know the objectives of studying pharmaceutical technology. So what are the objectives of this course? After completing the whole course content, you will be able to learn and explore the development of novel pharmaceutical processes, you would be able to learn an interdisciplinary approach in pharmaceutical process research and development. The third objective is to learn several pharmaceutical technology aspects, particularly the manufacture of various pharmaceutical dosage form in relation to physical pharmacy, applied biopharmaceutics, and pharmacokinetics. And the fourth objective is to design and develop the novel drug delivery systems and devices. So what we are going to study in the course throughout the semester. Following are the contents of the course that are divided into 17 topics. In topic number 1, in lecture number 1, that is of today, introduction to the course and overview of pharmaceutical technology would be done. In topic number 2, lecture number 2, principles of pharmaceutical formulation, dosage form design would be taught. In lecture number 3, we would learn a need for dosage forms. In topic 4, we would actually be learning pre-formulation studies. In topic number 5, polymers used in drug delivery systems would be explained. In 6th lecture, novel drug delivery system would be explained with an introduction to the new drug delivery systems. In 7th class, you would learn the sustained and controlled release drug delivery systems. In class 8, you would be able to learn the microencapsulation techniques. There are various microencapsulation techniques available, but we would actually be focusing on these 4 techniques that are coagulation, phase separation, solvent evaporation, interfacial polymerization, and spray drying. In lecture number 9, we would be learning about the developmental aspects of matrix and reservoir systems. In class 10, we would be learning about the advanced granulation technology design and practices. You might have learned granulation technology in your 4th year in industrial pharmacy, but here we would be learning about the advances in granulation technology, and we would be actually learning each granulation technology process in respect to advancement. In lecture 11, that is actually a subunit of this lecture 10, we would actually be learning about spray drying granulation technology. In class 12, we would be learning roller compaction technology. Extrusion and spheronization technique would be explained in class 13. Single pot processing would be explained in class 14. Rapid release granulation technology would be explained in class 15. And in class 16, we will be learning about particle coating by centrifugation granulation technology. And in class 17, we will be learning about product formulation. Now you might think that how only 17 lectures would be sufficient enough to complete the contents. Well, in practice, these are not 17 lectures but 17 topics. Each of the lecture would be subdivided into a minimum of 3 parts, and it may further be increased depending upon the length of the topic. What reference books you will be using in this class? You will be using different reference books. These books will be available through your HULMS via a OneDrive link. You are advised to download these books on your laptops or desktops. Whenever a lecture is completed and whenever an announcement is displayed on your HULMS, you would be required to refer a particular book quoted in the lecture or in the announcement. So let's proceed to the introduction to pharmaceutical technology. Pharmaceutical sciences and technology have progressed enormously in the recent years, and that significant advances in therapeutics and understanding of the need of optimized drug delivery in the body have brought about an increased awareness of the valuable role played by the dosage form in therapy. In turn, this has resulted in an increased sophistication and level of expertise in design, development, manufacture, testing, and regulation of drugs and dosage forms. This fact is as true today as it was in late 20th century and the early 21st century. And perhaps more so given the increasing emphasis being placed on the discovery, development, and use of large molecular entities as therapeutics and diagnostic agents. Pharmaceutical technology aims to providing the skills to sustain an external industrial environment and endeavor themselves in the research. How would you define pharmaceutical technology? Pharmaceutical technology is the application of scientific knowledge or technology to pharmacy, pharmacology, and pharmaceutical industry. It includes methods, techniques, and instrumentation in the manufacture, preparation, compounding, dispensing, packaging, and storing of the drugs and other preparations used in diagnostic and determinative procedures and in the treatment of patients. Let's see some examples of the technology in innovation and advancements. Traditional medicines versus personalized medicines. The world of therapeutics is now more focused on precision medicine concept. So the trend is shifting from traditional medicine to medicine or conventional medicine to personalized medicines or precision medicines. The medicines of the present are actually same for all kind of population that actually uses one size fits for all approach. Whereas medicines of the future are actually personalized treatments or personalized treatment options where treatment strategy is based on patient's unique genetic profile. Molecular testing is carried out on the affected patients and specialized personalized treatments or treatment options are used to cure the diseases. Pharmaceutical technology have played a vital role in the shift from conventional therapeutic options to modified drug delivery systems. Here you can see an example of insulin. Conventional insulin is available as an injectable dosage form. Although there are various types of insulins that are available in the market, which is again a result of advancement in delivery systems. Currently, rapid-acting, regular or short-acting, intermediate-acting, long-acting and ultra-long-acting insulins are available in the market and intended to be administered via injection only. The administration via syringe leads to incorrect administration of doses in majority of the instances. The advancement in technology had led to the development of insulin pens that are more accurate than syringes for doses. In 2015, an inhaled insulin product with the name Afrezza became available in the United States. Afrezza is a rapid-acting inhaled insulin that is administered at the beginning of each meal and can be used by adults with type 1 and type 2 diabetes. This gives ease of administration for patients as the injectable route is totally eliminated. Nonos is a concept of a smart insulin watch that frees the patient from needle injection. It works based on the principle of thermophoresis, where insulin is released and absorbed by the skin. Current method of insulin injection with a needle requires the use of complex apparatus. Not to mention, it causes pain and psychological discomfort. There is also possibility of allergic reaction due to high concentration of insulin. This new innovative method offers an innovative and convenient solution of insulin absorption. Insulin is injected into the patient through absorption onto the wrist skin. In principle, where an infrared lamp applies heat to the porous titanium nickel, which is also called as nitriol-shaped memory alloy, that is transplanted in a living body. In a living body, insulin inside porous body can be absorbed onto the skin tissue through capillary phenomena. This process actually simplifies and minimizes potential mistakes from self-administered injections, since the device can actually detect patient conditions and blood sugar levels as well. In 2015, FDA approved first drug product combined with digestive sensor. William Gibson once said, and I quote, that future is already here. It's just not very evenly distributed. And that is certainly true for digital health system. The Japanese pharmaceutical organization Otsuka and Proteus Digital Health combined a digestible sensor with a type of drug against mental illness. With the patient's consent, the indigestible sensor communicates with the wearable sensor patch if the drug is taken. Then the information is transmitted to a smartphone or tablet of the curator or the patient himself. Let's watch this video for the better understanding of the concept.
The U.S. Food and Drug Administration has given the green light to a new medication that will track whether a patient is following their prescription. Some ethicists, though, say it's a tough pill to swallow. Tashana Reed with more on what is being called a digital pill. Tashana.
Well, Heather, I'm going to go right into explaining exactly how this works. So what this is it's a medication that is embedded with a sensor, and this is something that the patient would take and ingest orally. And so the sensor contains a copper and magnesium and silicon part and generates an electrical signal when it comes into contact with stomach fluid. So as the person takes it, it hits your stomach juices, and that is when within a few minutes a signal will be sent out, and that signal is detected by a patch that is worn above the left rib cage. So that patch then sends the date and time of when that pill was ingested to the patient's cell phone app. And so that's what's very interesting here. This is an app that the patient would download on their phone, and I'll show you an image here because from here the patient can tell the application what their mood is at the time, when they've actually rested, and also they can have this information sent to not only their doctor but people that are part of their medical team or support team, so maybe family members or support workers. So this technology was created by a company called Proteus Digital Health, and they teamed up with a pharmaceutical company, and in particular this approval comes for a medication that is for patients with schizophrenia, depression, and bipolar disorder. And doctors are hoping that this will ensure these particular patients to take their medications when they are supposed to, because many people dealing with mental illness don't necessarily take their medications because of some of the side effects. So that is what doctors are hoping for, but many experts saying that this approval is a huge step forward when it comes to digital health.
Well, in what way? I can certainly understand the non-compliance issue, hopefully helping patients who have some concerns there. But how else do doctors see this as being more advantageous?
Well, one of the biggest challenges doctors say is the number of people who don't take their medications accordingly and how they are prescribed to. And what ends up happening is people get sicker. And so what that means is it's costing the United States $100 billion a year. That's what experts say is the final total because of further treatment or hospitalization when people aren't correctly taking their medication. Now, experts also say this technology can help in a vast number of areas, including for people who potentially forget to take their medication. They've also suggested patients who've had surgery and need to take opioids to make sure that they are also taking those appropriately. So there are many other medications similar to this and technologies that are being developed. This approval means that a lot of those can potentially go forward. But as you mentioned, a number of ethical issues as well. Many people saying that this seems a bit Big Brother, having all of this monitoring around someone taking medication.
With biomedical engineering, testing drugs on humans or animals in the framework of costly and lengthy clinical trials are the past. Organs on chips concept is the new type. Imagine that the characteristic of living organisms are so perfectly simulated that the clinical trial can be carried out in less time with less money and still amazing outcomes can be obtained. Let's watch this video of the method.
Imagine a very tiny human, so small its brain and other organs are the size of rice. Now imagine this microhuman testing everything from prescription drugs to toxins and chemicals. A version of this microhuman is real.
The idea is to create a human on a chip.
Its brain is being built here at Vanderbilt, led by Professor of Biomedical Engineering, Physiology, and Physics John Wixwow and his team at the Vanderbilt Institute for Integrative Biosystems Research and Education.
You get cells that are directly derived from humans, and you grow them up to create organs.
Leading to much faster and more accurate testing.
It's clear that organs on a chip will help streamline the pharmaceutical pipeline, and people should benefit from that without having to have drugs be pulled from the market because of unanticipated side effects.
Vanderbilt and fellow institutions are partnering in a multi-million-dollar program sponsored by the National Institutes of Health and other government agencies. Today, Vanderbilt scientists are growing a version of a brain.
This is our neurovascular unit on a chip, or we can just say brain on a chip.
They're specifically testing the blood-brain barrier.
To show when you deliver a drug, does it get to the brain or does it not? Does it get to the brain in the form that you want?
Other institutions are building hearts, kidneys, livers, and more.
It's actually a great deal of fun. It's a challenge.
Another key contribution by Vanderbilt?
We have all the hardware to keep different organs alive.
And ready for study.
Microclinical analyzers to see what the cells are doing. Perfusion controllers that keep the cells alive for weeks or months or more.
This device is a mini laboratory.
Retrieve sample from that organ on a chip, send it directly to our sensors here, and allow us to determine if a major change has happened due to that drug.
And incredibly complex because of the minute size.
One of the challenges in the organs on a chip business is that when you're building a millihuman or a microhuman, you don't have very much fluid to work with.
That's where this invention comes in. Also created by Vanderbilt, it tests exceptionally small amounts of fluid taken from the organs on a chip. Literally dots.
I think this is an extremely exciting time for both science and engineering.
A key element to this project was born by Vanderbilt's youngest scientists.
Some undergraduates and even high school students working with us came up with some very clever ideas to make very small pumps and valves that were very low cost. And that has led to a complete watershed of projects.
Now partner institutions are using Vanderbilt's devices. Next steps?
The project in front of us is to have 4 organs merged to make probably about a 10 microhuman human on a chip.
To see how multiple organs react from a certain drug or toxin, mimicking a real human body.
From my point of view, the trick is to get the hardware smaller and cheaper so that you can afford to put hundreds or thousands or tens of thousands of copies of it into toxicology and pharmacology labs.
Revolutionizing the future of medicine and our health.
This method is called the silico trial method. It is basically an individualized computer simulation used in the development or regulatory evaluation of a medicational product, device, or intervention. While completely simulated trials are not feasible with this current technology and understanding of the biology, its development would be expected to have a major benefit on current in vivo clinical trials. Well, this is not the limit. There are hundreds of advancements in pharmaceutical technology that have led to the more developed and advanced practices of pharmacy in every field, whether it may be hospital pharmacy, community pharmacy, industrial pharmacy, or even research. Today, if you have any questions, you may use the HULMS portal or simply write an email to me. Thank you for patiently listening to the lecture. Take a very good care of yourself and everyone around you. Allah Hafiz.